Good evening, everyone. Welcome to today's special online webinar on the topic amino acid and short peptide based cell assembled materials conducted by the Department of Chemistry of Mrinalini Datta Mavavidyapith. Before starting the webinar, I would like to ask the participating members to mute yourself to run the webinar smoothly. You all are free to ask your questions and doubts in the chat box. And after the lecture gets over, I will read out your questions on behalf of you. Today's speaker is Dr. Jayanta Nanda, Assistant Professor of Department of Chemistry of University of North Bengal. I would like to request Dr. Prabuddha Bhattacharya, Lecture Coordinator, Head of the Department of Chemistry, Mrinalini Datta Mavavidyapith, to speak a few lines on today's webinar.
Thank you, Vishwajit. So, good afternoon, good evening to everyone present. On behalf of the Department of Chemistry, Mrinalini Mahavidyapith, I extend a very warm welcome to all of you. Thank you for showing your interest in our ongoing special lecture series. Today, we feel extremely honored to welcome Dr. Jayanta Nando, Assistant Professor from the Department of Chemistry, University of North Bengal. We express our sincere gratitude to him for accepting our proposal to deliver an online lecture to our participants. Thank you, Dr. Nanda, and on behalf of the organizing committee, I welcome you once again. Dr. Nanda is a prolific researcher in the field of peptide based supramolecular chemistry and biomimetic materials. From what I understand in his today's lecture on amino acid and short peptide based self assembled materials, Dr. Nanda will be sharing his valuable insights on how simple amino acids, owing to their chirality, hydrogen bonding tendencies, and diverse side chain functionalities, act as fundamental building blocks capable of forming highly ordered supramolecular structures. We are all aware that short peptides have emerged as powerful minimalist platforms for designing biomimetic materials, drug delivery systems, catalytic scaffolds, and responsive nanostructures. In this context, Dr. Nanda's lecture will also highlight the fascinating world of short peptide based assemblies, where small peptide motifs exhibit remarkable structural precision and tunability. With this brief introduction, let me welcome all our participants once again. I am sure we will all be having an intriguing session in the next couple of hours. Let me hand over to Vishwajit. Thank you.
Thank you, sir. Now I would like to request Dr. Sunanda De, Faculty Member, Department of Chemistry, Mrinalini Datta Mavavidyapith, to kindly introduce our today's esteemed speaker and initiate the lecture.
It is a great pleasure to introduce Dr. Jonathan Nando, Assistant Professor in the Department of Chemistry at University of North Bengal. With a research career spanning in India and abroad, he brings a rich background in supramolecular and peptide-based material science. His education includes an MSc from University of Calcutta and PhD from ICS Kolkata, then followed by postdoctoral work at Ben Gurion University of the Negev, Israel, and Radboud University, Netherlands. After that, he has started his independent research career as a DST Inspire faculty in IIEST Sipur, Kolkata. Then he joined as assistant professor in the Department of Chemistry at University of North Bengal. Dr. Nando leads the self-assembling peptide chemistry laboratory, where his group focuses on the design and synthesis of short peptide derivatives for biomimetic functions and their self-assembly or aggregation behavior, particularly gelation, crystallization, and functional supramolecular phenomena. In essence, his work sites at the interface of organic chemistry and nanostructured materials and life-inspired self-assembly. Dr. Nando has published over 40 peer-reviewed articles accumulating more than 2,000 citations in the field of supramolecular peptide chemistry. And we are honored to welcome Dr. Nando as our speaker, whose pioneering work on peptide self-assembly and functional nanomaterials promises to inspire the fresh insights and discussion. I welcome Dr. Nando.
And thank you, Dr. De. Thank you, Dr. Bhattacharya, for the invitation and the introduction. I want to correct it. I have published 33 paper, not 40. So anyway, so just let me share my slide. Okay, my screen is visible. Okay, okay. So very good evening to everybody, to all the participants, dear students, and the colleagues and the faculty member of the Mrinalini Datta Mahavidyalaya. So today I will talk about amino acid and short peptide based self-assembled material. So I will start my lecture with one picture. So what is this picture? This picture are the Lego. This picture are the different building block. And I hope some of the participants had played using this Lego. So using this Lego, we can build wall, we can build house. And when we give this Lego packet to kids, so they enjoy it and they use their creativity to building different structure. And if you see the Lego, so if you just press it, so that will be jump. So we can build up different structure. Like you can see one part is add here, and from the back side there is another part. So when you press it, we can build different kind of wall. Okay, different kind of train, different kind of house. So in nature also builds different kind of structure. So what are the structure? The structure of the nucleic acid, the structure of the carbohydrate, the structure of the proteins, lipid. So these are the different biopolymer. So nature build this biopolymer from very small functional building block. What are those? These are the amino acid, these are the sugar, these are the nucleic acid, DNA or RNA. So from this building block, the nature can build different functional biopolymer, which is doing different kind of work, functionality in our cell. Most of us from the chemistry background, and we all know what is synthon. Synthon term was introduced by E.J. Corey in 1967, that the equivalence of the, suppose we want to synthesize this molecule, so what will be the building block, what will be the starting material, equivalence. So this term is called the synthon. So similarly, Professor Gautam Desiraju from our India, he introduced the term supramolecular synthon. So different biomolecule, different building block, they when they crystallize, they use different kind of intermolecular interaction, and depending upon their structure and function, different crystal structure is formed. So he introduced this concept in his paper, Supramolecular Synthon in Crystal Engineering, a New Organic Synthesis. So what are the non-covalent interaction? Non-covalent interaction, the hydrogen bonding interaction. It can be the aromatic interaction, ionic interaction, hydrophobic interaction, and others. So if you see, the atoms are connected to form molecule by covalent bond. And when this molecule are formed, this molecule uses different kind of non-covalent interaction forming supramolecular assembly. And in this supramolecular assembly, some new functionality emergence of new functionality, which was not present in the molecule, So small building block, they are connected by the different non-covalent interaction and forming the supramolecular assemblies. And in supramolecular assemblies, in general, we find some new functional property. In our lab, we work on the amino acid. So this is the general structure of the amino acid. This is the general structure of the amino acid. Amino and acid. So this is the amine part and this is the acid part. So usually we call it alpha amino acid. Why this is called alpha amino acid? Because the amine and carboxylic acid, the priority of the functional group, carboxylic acid is higher, right? So carboxylic, just a minute. Sorry. So carboxylic is priority is higher. So this functional group attached to this carbon. So this is the alpha carbon and amine group is attached at here. So this is the alpha amino acid. And in alpha amino acid structure, only variant is R. Depending upon the R, there are the different amino acids. So most of the amino acid R is varied. So here you can see this is the aromatic functional group like phenylalanine, tryptophan, tyrosine, histidine. It can be basic, it can be anionic, it can be nucleophilic, it can be aliphatic. So based on the different amino acid, if you synthesize a different peptide, their property, their structure, their function will be different. So these are the building block. These are the Lego in the synthesis. It is in the nature, it is if you synthesize in the laboratory, this functionality are different, and using this building block you can synthesize different short peptide and they will have different property and function. So this is the alpha amino acid, and if you see I have drawn another two amino acid. What is this amino acid? This is the beta alanine. Actually this is the beta amino acid. So this is the alpha carbon and this is the beta carbon. So in beta carbon, the NH2 group is there. This is the beta alanine. This is the isomer of the alanine. Usually if R is equal to CH3, this is the alanine amino acid, and beta alanine is the isomeric molecule of the naturally occurring alanine. So beta alanine also present in the nature. And secondly, I have drawn another structure. This is the GABA. GABA full form is gamma amino butyric acid. So this is the alpha position, beta and gamma. So gamma amino butyric acid. How many carbon is there? 1, 2, 3, 4. 4 means butyric acid. So gamma amino butyric acid. So not only alpha amino acid, if you use different kind of beta amino acid, gamma amino acid, omega amino acid, you can build different kind of peptide and it will provide different kind of nanostructure and it will exhibit different kind of function in the self-assembled condition. So I will talk about few minutes on the amino acid, from amino acid, how peptide bond are formed. So I have assigned at this is the amino acid A and this is the amino acid B. If you see, their side chain R group are different. Okay. So if I ask before the peptide bond formation, how we can synthesize ester bond from a carboxylic acid and from a alcohol. It is easy. So if you take acid and alcohol, put a catalytic amount of acid and reflux, we'll get the ester bond. So using this approach, we can synthesize the peptide bond. So when we take acid and amine and reflux or taken in a solvent and reflux, what will happen? So basically, we will not get a peptide bond like the scheme shown at here, Okay, so how the synthesis occur at here? Because this is the carboxylic acid and this is the amine. This is the acid and amine. So when you took this, so it will form salt, right? So if you think about the peptide bond formation, so NH2 group will attack at here, the OH group will be removed. But if you think the OH group is a bad leaving group, so for the peptide synthesis, you have to make the OH group as a good leaving group with the help of different peptide coupling agent. different peptide coupling agents. So what will happen? The NH2 group attack at here, and the modified OH group, which is turns into the good living group, this group will be removed, and the peptide bond will be formed. Peptide bond will be formed. So this is the general method. So if I take A and B, two amino acid, so we can expect the AB molecule will be formed. The AB molecule will be formed. Whether any other possibility? Yes, there is other possibility. What are the possibility? The A molecule can self-condense. If you think about only dipeptide formation, A molecule can self-condense and form AA. B molecule can self-condense, it can form BB. And similarly, if this molecule condense in reverse way, it can form the BA molecule. So our target was to getting AB peptide from AB. Okay. So if you think about the 100% reaction occur, so ultimately we'll get around 25%. Right. If I go for the next step, so again the we'll get lot of other product which will decrease the overall yield. So for that purpose, we have to introduce some protecting group at the N terminus, and we have to introduce some protecting group at the C terminus to make sure the reaction take place in this two end in presence of peptide coupling agent. Okay, so peptide coupling agent usually use DCC. Usually we use HBTU. DCC is the first peptide coupling agent came in the market, and after that different water soluble peptide coupling agent also introduced. And again, if you want to synthesize another amino acid after AB, if you want to introduce C amino acid at here, so either you have to remove the C terminal protecting group and introduce C amino acid like this scheme, or we can remove the protecting group at the N terminal and couple with the C and the AB, and we can prepare in this way depending upon our requirement. So these methods are basically solution phase method. In solution phase method, after each coupling, you have to purify. Otherwise, if there is some impurity, it can interfere in the next step. But if you synthesize large peptide, in the solution phase methodology, what will happen after 4, 5, 6 amino acid? So the peptide will be too hydrophobic, it will not remain in the solution. It sometimes precipitated in the solution. So the extent of reaction will be reduced, because sometimes we written R group at here, but this R group can have if there is methyl, so there is methyl, but if there is glutamic acid or aspartic acid, so carboxylic acid group should be protected. So these N-terminally protected, C-terminally protected, as well as the side chain protected peptide became too hydrophobic to do reaction in the solution. So for that purpose, solid phase synthesis came in the market. Merwin developed this approach, and in the solid phase approach, you have to design your sequence, what you want to synthesize, and the synthesis is very easy, because after each step, the excess reagent can be washed out. So in the peptide synthesis, what we have to think about: number one, protecting group, N-terminal protecting group, C-terminal protecting group. We have to use proper coupling agent, and the methodology, whether it is solution phase methodology or it is solid phase methodology. So after synthesizing the peptide, what you have to do? You have to purify. You have to purify using, normally we do in the silica gel column, we purify the compound, and if the compound is N-terminal or C-terminal is reprotected, that is NH2 and carboxylic acid free, we used to do purification in the HPLC. So what we do using this peptide? So the question is, why we will study the peptide based material? Because we are living species in our body, in our system. One of the major biopolymer is peptide. So to understand the different living system and their way of function, we know different enzyme, different protein do several work in our system. So how they are synthesized? We took food and we took protein in our food, and when you take a food, our body, our enzyme, digestive enzyme, break into the corresponding amino acid, and depending upon body's requirement, the different protein is formed. So this protein do their job, and after doing their job, either it will be decomposed or it will be function in such a way that it will be removed from our body. Okay, so these are, so to know how the living system work and their function, we have to know the different peptide-based material, how they are synthesized in the laboratory, how they are synthesized in the cell, and other thing. And moreover, if you see, if you have some old man, that is grandfather, grandmother, even parents, sometimes they cannot remember who came today in our home, or if you face somebody is writing but hand is shaking. So these are some kind of disease. These are disease, neurodegenerative disease. Basically, these peptide, there are few peptide. These peptide was soluble in nature, but with time they are aggregated, misfolded, and they form insoluble aggregate, and these insoluble fiber are deposited our nervous system, our brain. So the some of the function we cannot do properly. So these are the disease. Which are the disease? These diseases are the Alzheimer disease, Parkinson disease, Huntington disease. So these are the different kind of disease. So these are occurred due to the misfolding of the protein which was present in our body. So if we know how this misfolding occur, how we can stop the fibril formation, we can prevent, we can basically prevent the Alzheimer disease or some other disease. Okay. So for that purpose, we have to know the protein and their function. And thirdly, peptide-based biomaterial for the mankind. I have shown another cartoon ad here. So you can synthesize different peptide based on your requirement. And this peptide-based material, due to its origin, they are biocompatible, they are biodegradable. And this peptide-based material can be used in the drug delivery, 3D fabrication, bone regeneration, wound healing, tissue engineering, in a different way. In the modern days, this self-assembled peptide-based material used in the various aspects. I want to show another image which is based on the peptide. So what are those? These are the spider silk. This is very common. Like in the evening, you can see some spider is building the net. Okay. So these are made of peptide. Even these peptide have a high strength. And here I have shown the image of silk. So all are based on the peptide. All are based on the peptide. So this peptide-based material you can use various kind of way. So in our laboratory, we mainly work on gel material. We also work on the short peptide-based crystal. So when we synthesize some peptide, what we'll do, we dissolve in a particular solvent, like depending upon their functionality. Sometimes we study in water. If the peptide N terminus and C terminus is deprotected, we study sometimes in water. Sometimes protected compound we study in the organic solvent. So we heat it under control way and make a saturated solution. So when we make a solution and upon cooling, there is a three option. One option, it can precipitate, which generally happens, it precipitate, or in other two way are the crystal formation or the gel formation. So depending upon if it is formed gel, so you can see it will stop the flow of the solvent. It will stop the flow of the solvent. So these are the molecule using different kind of trigger. I will coming later. So when you put some kind of trigger, so they are self-assembled, and when they're self-assembled, they are forming fibril. And this fibril stop the flow of the solvent where you have studied. And if you invert the test tube, you will see the solvent is not fall. So it actually immobilize the solvent. So what kind of trigger you can use? So normally heating cooling cycle, if you heat it, make it monomer, and sometimes you can get the gel. This is a commonly used approach. Otherwise, you can change the pH. You can change the pH. You can use some kind of sonication energy. You can use some salt concentration, increase or decrease. Okay, so by this physical stimuli, you can trigger the self-assembly, and these molecules are self-assembled with the help of different non-covalent interaction like pi-pi stacking, hydrogen bonding interaction, and they are forming fibril. And this fibril, when they immobilize the solvent, we are getting the gel. So gel can be classified in the different way based on the source. Based on the source, number one, whether it is natural, like aloe vera gel. This is a natural gel. But when you are synthesizing in the laboratory, so this is the artificial. And in the laboratory, you can synthesize some small molecule and you can synthesize large molecule, and some molecule are cross-linked. Some molecule are cross-linked, so these are polymeric gel. And some small molecule, when you tune the solvent medium or use some sonication, or if you use some trigger, so this small molecule self-assemble and they form supramolecular gel. Basically, small molecule based gel are physical gel. Okay. And what is the medium? If you put organic solvent, so this is the organogel. If you put water, so this is the hydrogel. So another type of gel it is called, but not important that much. So that is called aerogel or gerogel. Suppose you have prepare a gel, and if you remove the solvent, so what will happen? So you will see some fiber is there. So when there is fiber, so there is no solvent, so it is called sometimes the gerogel. Few people called it aerogel. Why? Because there is nothing, so there is air is there, so it is called aerogel. Basically, ignore this part. It can be organogel or it can be the hydrogel. Depending upon the source, it can be either natural or artificial. And small molecule based gel are the supramolecular gel or physical gel. So in this presentation, I will talk about mainly two systems. Number one is single amino acid based self-assembled system. Okay, and second one is design and development of artificial helical system and their helicity tuning. So if you see the peptide-based fibril formation, so these peptide-based fibril formation are due to the different kind of non-covalent interaction. What are those? Like hydrophobic interaction, hydrogen bonding interaction, pi-pi stacking, ioning. Among these interaction, pi-pi stacking interaction is very important. So I have shown the picture of Professor Ehud Gazit. He's from Tel Aviv University, Israel. He did lot of work on the short peptide based self-assembled system. He postulated in 2004 that the role of pi-pi stacking interaction in the amyloid fibril formation. So if you see the sequence, this is the A beta amyloid sequence 42 residue, which is responsible for the amyloid fibril formation, amyloid disease. So he studied different fragments of this A beta amyloid sequence, and they use the minimal approach. They took the shortest fragment which is responsible for the fibril formation. So initially they studied the long peptide. After that, they studied this red portion. This sequence is one letter sequence is KLVFF. KLVFF, this is the 7-mer peptide. And not only him, other group also studied. And after that, they found that KLVFF it is forming fibril like the original sequence. And after that, they found the FF, which is phenylalanine. They are also forming fibril or nanostructure like the A beta amyloid peptide and toxic. So they have also worked on the dipeptide, Phi dipeptide, N terminal is free and C terminal is free. And they first time reported that achiral dipeptide can also form the nanotube. Okay, so coming in the point, so later they found that itself phenylalanine, they can also form toxic fiber like the A beta amyloid peptide. So they have published a Nature Biochem in 2012 where they first time connected the phenylalanine self-assembly with phenylketonuria. What is phenylketonuria? Phenylketonuria is a inborn disease, deformation, where if the mother, if the pregnant woman had in her body, the percentage of the phenylalanine, accumulation of phenylalanine is higher. Phenylalanine is a essential amino acid. We cannot prepare in our body. We have to take through the food. Okay, so like different seeds, meat, nuts. So it contains high amount soybean. It contains high amount of phenylalanine. So when we take phenylalanine with the through food in our body, there is an enzyme that is called phenylalanine hydroxylase. What they do? So it transforms phenylalanine to the tyrosine. But due to some gene mutation, some disorder in the mother, so what happened? The phenylalanine to tyrosine conversion rate is poor or very less. So phenylalanine tyrosine, from phenylalanine tyrosine formation is reduced. And we know from tyrosine there is a formation of dopa. From dopa there is formation of dopamine and other very few important biological molecule which is needed for the development of any human being. Okay, so when phenylalanine accumulation is higher in our body, so this phenylalanine they form fibril, and from mother, so this phenylalanine content, that is phenylalanine, comes into the baby, and this baby have some deformation or some not deformation, some mental retardation. Okay, so what youth gadget done? So he connected the phenylalanine, what we take for the as essential amino acid. So this phenylalanine can form toxic fiber like the native peptide. Okay, so I also work on the phenylalanine. In our case, our approach was, so if you see the phenylalanine, I'm coming back. Okay, if you see the phenylalanine, so this is the aromatic ring, and we know from our previous classes that like BSc or MSc classes or 11, 12 class, if you put some electron donating group or electron withdrawing group, it can influence the pi electron cloud. So we are working on some modified aromatic amino acid. So during that time, we thought about whether we can write a review on phenylalanine based fibrillar system, and we got an invitation from Chemcom, and we wrote a feature article on phenylalanine based fibrillar system. So in which condition we have summarized, in which condition phenylalanine form fibril. Okay. So if you change pH, ionic strain, solvent, temperature, in which condition phenylalanine forming fibril, and what are the condition, in which condition you can stop the fibril formation. So if you stop the fibril formation, it forming the non-toxic small aggregate. It has been observed that I find in different paper that if you self-assemble some drug, polyphenolic compound, metal ion, or cyclic ether nanoparticle with a phenylalanine, so it is basically stop the fibril formation. So if you're interested, you can read this review, how or in which condition phenylalanine form fibril, and in which condition, or in presence of science, the blessings of science, in which condition the fibril formation is stopped. So basically, in phenylalanine-based system or aromatic amino acid-based system, our motivation is we find few paper that time in literature that if you introduce some group, some electron withdrawing group or donating group, halogen, so it actually influence the self-assembly. So there is a sequence of the pentapeptide. In the pentapeptide, there is a phenylalanine ring, and Metrangolo, they studied when they put halogen group here, they find that the normal sequence, it is not forming gel, but when you put at least one halogen, they are forming gel. And another group, Nelson group, sorry, they have put halogen, and they find that incorporation of the halogen actually assisted in the better self-assembly process. We find another paper that nitration of tyrosine 10 critically enhance the A-beta, amyloid beta aggregate and plaque formation. So the sequence I have shown previously, that is the A-beta amyloid sequence, and in the number 10 position there was a tyrosine, and due to the nitration, this group, Henka and Coworker, they reported that when the nitration occurred at the tyrosine, there is a better aggregation process. So we thought about, so in our system or in the laboratory, we can find tyrosine, tryptophan, phenylalanine. So we took tyrosine and phenylalanine, and we think about whether we can do simple nitration by concentrated sulfuric acid and nitric acid, whether it can form nitrobenzene or nitration in this ring. And our plan was, so if we introduce this aromatic nitroamino acid in the peptide sequence, how it will influence the self-assembly process. So for that purpose, we first synthesize the 4-nitrophenylalanine. 4-nitrophenylalanine, what we have done, we have taken phenylalanine, and we have used concentrated sulfuric acid and nitric acid, and nitration occur at the 4th position. We have crystallized the 4-nitrophenylalanine. When we solve the crystal, we find that it came with an water. So in our lab, we want to synthesize, we want to introduce this amino acid in the peptide sequence. So for that purpose, we want to first we want to check the NMR, and also want to check dissolve this compound in some other organic solvent, basically DMS or DMF in the solvent. So when we dissolve this compound in DMF, we find that 4-nitrophenylalanine forming a self-supported gel, and we find that this is this forming gel in a very lower concentration. That is, it is called the minimum gelation concentration. When you have a very less amount of compound gelify the organic solvent, so its efficiency is high. So in another case, we find that it is also having the water molecule. Okay, so we thought about water and DMS. So these are miscible solvent. So when we in the water we are getting crystal, and in the DMSO we are getting the what is called the gel. So we think about when we mix it, what will happen? We find that in DMSO it is forming gel up to this is the pure, this is the 90%, and this is the 80%, and the rest of the cases it is forming the crystal. We started the UV visible spectroscopy, and we find that in case of the DMSO, there is a new peak generated around 480. But when we increase the percentage of water, this peak actually vanishes. This peak actually vanishes, and it forming the less color solution. So basically, here DMSO is electron rich solvent, and 4-nitrophenylalanine is electron deficient. So they are forming charge transfer complex formation, and they also forming gel, they also forming fibril. So basically you can see here that depending upon the solvent composition, if the percentage of water is less than 20%, is forming the charge transfer gel, and if it is percentage is higher than 20%, is forming the crystal. Later, we have introduced histidine, phenylalanine, tyrosine within the gel matrix, and we find that the color became more intense, and it indicates that in presence of this amino acid, they are forming charge transfer complex. And in the second example, here you can see this is the tyrosine, and we have done the nitration. So we got the 3-nitrotyrosine. 3-nitrotyrosine, it is forming in our body in presence of reactive nitrogenating species. So if somebody asks why the color of urine is yellow, so we usually say that it is bilirubin or biliverdin. Apart from that, the third responsible compound is tyrosine, 3-nitrotyrosine. So what we have done, we have attached ABMOC group, and we have studied the gelation of the ABMOC nitrotyrosine, and we find that it forming gel in the wide range, like from 5 to 8.5. Another interesting observation you can see here, the color. So around 5.5, this is less color, but around 8.5, this is more intense color. So these color changes basically occur due to the deprotonation of the phenol group. So if you see this molecule, this is the orthonitrophenol. So when you increase the pH, so you can see the more the phenol group is deprotonated, and the more conjugation will occur, and the coloration will increase. And this gel, we find that this is temperature responsive, and this is also mechanical stress responsive. So if you take the gel and if you shake it, so it will transform into the solution, and again if you rest it, it will again back to the gel. This is called the thixotropic property. So we have here studied the two system, nitrophenylalanine and 3-nitrotyrosine, which is by preparing the modification. Here another system we will discuss. This is the tyrosine molecule. Tyrosine molecule in DMSO, it is forming the gel, and we have studied the charge transfer complex formation with an electron deficient molecule, what is called the DDQ. So when we study this gel material, it has been found that hydrogen bonding interaction, bypass stacking interaction are responsible for the gel formation. But when we introduce the DDQ, this is the electron rich system, and this is the electron deficient system. So they are forming the charge transfer complex formation, and new color format here. And when you check their nanoscale morphology, initially this is the fibril, and the fibrillar morphology was damaged at here. So quickly I will talk about another system, that is the ABMOC phenylalanine. We know that R group is unique. This side chain is unique. If you change the side chain, its property will be changed. So there was a report in Soft Matter 2011 that this compound ABMOC phenylalanine, they forming hydrogel, and they are also exhibiting the antimicrobial activity. There is another paper in Soft Matter 2015, and they claim that the phenylalanine side chain and the buffer and the ABMOC group is important. So we only concentrated on the ABMOC side chain. So what we have done, we have removed the side chains aromaticity. So it is forming the another amino acid. This is the cyclohexylalanine. In another case, we increase the flexibility. Here one CH2, but you can see here there is a 2 CH2. And another case, we have removed the flexibility. So this is CHA, cyclohexylalanine. This is the phenyl glycine, and this is the homophenylalanine. And we want to see whether what is the role. So we find that in presence of ABMO group, all these compound, all these analog, they are forming hydrogel. And I have not shown the data, and we find that the phenyl glycine out of this new compound, phenyl glycine is better antimicrobial activity towards than the phenylalanine. And so what we have done at here, we have done the side chain modification, and we also find, we discuss the role of the side chain, and we find in some cases this is the better antimicrobial, exhibiting better antimicrobial activity. So in the second part, I will talk about the helical structure, helical or chiral structure. So chirality is everywhere, and it can be observed in various hierarchical level from subatomic and molecular and supramolecular to the microscopic scale. So I have given some image. So neutrino, the chiral molecule, this is the biopolymer, this is the plant, this is the galaxy, this is the tobacco mosaic virus, the helical horn, the flower, snail cell, this is the seed cell, this is the hemisphere. So everywhere there is some chirality or chiral architecture present in our nature. Actually, we are living in the chiral environment. The biopolymer which was present in our body, all are chiral. So we want to synthesize, we want to mimic some chiral helical structure. So what we have done, we have synthesized naphthalimide conjugated dipeptide. So naphthalic anhydride reflux with amine, and it is from naphthalimide. Okay. So in our design, we want to keep naphthalimide to introduce another group, another aromatic group than ABMO group, which can assist in the self-assembly or gelation. Moreover, this naphthalimide is a fluorescent in the visible region, and the pi stacking interaction help in the gelation. And in our peptide design, we took 2 amino acids. This one is the achiral amino acid, and this is the chiral amino acid. So fluorescent part will be come from the naphthalimide, and the peptide part, the biocompatibility, will come from the peptide. So when we synthesize the first molecule, that is the beta alanine, N is equal to 2 beta alanine. So this compound, just a minute. So we observe that it is forming gel in the DMSO water, and when we check the morphology, it is forming the left-handed helical nanofiber. We thought about whether we can tune the helicity of the nanofiber. This helicity tuning is observed in the nature. The bDNA, this is the right-handed DNA, and under disease condition, it actually transformed to the zDNA. So we thought about whether we can, by some tricks, we can tune the helicity of the nanofiber. In our first design, what we have done, we have keep this molecule, and we only change the chirality of this alpha amino acid. So this is the alpha amino acid. And what we have done, we introduced the valine, L-valine. In another case, we have introduced the D-valine. And very structurally similar, we put another molecule. This is also alpha amino acid, but achiral. This is the 2-amino isobutyric acid, AIB. This is the molecule AIB. And when we study their self-assembly in the DMSO water medium, we find that NLB, NDB, they are forming gel, but the achiral molecule, these are not forming gel. So not only achilality, but also the strain in the molecule, it actually resisted the gel formation. Here we use the DMSO and water. Our compound are protected. C-terminal is protected. It is not dissolved in water. It is dissolved in DMSO. So DMSO, we dissolve in DMSO and we put water, and we observe that all these molecules are the chiral molecule, they are forming gel. Okay, and the achiral molecule, we don't find the gel. We have studied using the UV visible spectroscopy. We find that when you increase the water percentage, the lambda max is redshifted in the 3 cases. So this is an example of J type of aggregation. And we also studied the fluorescence property by varying the composition of the solvent, like the ratio of the water and the DMSO. We find in the lower percentage, the fluorescent intensity is less, but 50% there is an enhancement. So this is called aggregation induced enhancement in the emission property. So all of the compound exhibited the AIE property, and you can see interesting observation at here. In the series study, the NLB and NDB, they have a mirror image relationship, and NAA, it is passing through the zero. It is passing through the zero. Okay. So another thing you can see here. That this is the 0%. 0% means it is dissolved in the pure DMSO. All the compound having a chiral center, but when we put water 90%, so some signal is appear. So when we put water, so they are, though they are chiral, they are arranged in a chiral way. Supramolecular, they exhibiting supramolecular chirality. So when we study their morphology, we find that the NLB, it is forming the left-handed helical fiber, NDB, it is forming the right-handed helical nanofiber, and NAA, which is achiral, it is forming the flake-like morphology. So we make a TOC like this way. So in the molecule, all the parts are same, only the chirality part is different. So we can imagine like a radio channel. When you change the channel, so when you come to the L amino acid, when you come to the D amino acid, when to the achiral amino acid, we got the different kind of morphology. So we can tune the morphology of the self-assembled material by changing the chirality of the alpha amino acid. In the second word, we have tuned the helicity by achiral part. Usually, the helicity, or if you change, if you influence, so you need to add some chiral molecule, either chiral molecule or chiral solvent. But here we find that helicity change due to the achiral part. Okay, so I'm coming here in the synthesis. So we have taken the naphthalic anhydride and we have reflux with amino acid. So this is the achiral amino acid, and we attach the what is called the C-terminally protected isolation amino acid. Okay, by DCC HOBT coupling. Here, just concentrate on the red part. All the parts are same. In the red part, when n is equal to 1, we term as n1. N is equal to 1 means this is the glycine. When n is equal to 2, that is beta alanine. So similarly, when we increase the 3, 4, so the achiral part was increased. We have studied their gelation, and we find that up to, we started with the 3 millimolar concentration. In all cases, we find that in presence of 50% water, the N1, it is not forming gel, but for N2, N3, N5, from 50% water, it is forming gel. So we have studied the CD. And you can see this is a beautiful spectra, that in all the compound, a fixed chiral center is present. But what is the difference? Change in the achiral part. So if you see the N2 molecule, the peak in the negative direction. For N3, the peak in the positive direction. Again, from N3 to N4, it is again in the negative, for N5 it is in the positive. So the N2 and N4, which is the even number of methylene group, the CD signal in the downward, and N3 and N5, in which case the methylene unit are odd number, they exhibiting the CD signal in the positive direction, the first CD signal in the positive direction. So these clearly indicate that when you change the achiral part, methylene unit, so there is a clear odd even effect. So by changing the methylene unit, we can change the helicity. So we have studied the morphology at here. So in case of the N2, we are find this is the left-handed helical nanofiber, and in case of the N2 left, and for the N3, we are getting the right-handed helical nanofiber. So this is the summary. Like depending upon the methylene unit, odd or even, the helicity of the nanofiber is changed. So when N is equal to even, during the self-assembly, it exhibiting the left-handed helical nanofiber, and in another case, this is exhibiting the right-handed helical nanofiber. So this is the my last work. So last work I will present at here. So here, what we have find, the solvent composition dependent helicity tuning. Solvent composition. So which solvent we are using at here? DMSO and water. All are achiral. All are achiral. Okay. So previously we have discussed the relation of this molecule. This is the N2. N is equal to 2. This is the N2 molecule. That is N means beta-ala isoleucine. NMI beta-ala isoleucine. So we observe, when we studied the relation, we observed that within few minutes, within the gel matrix, something is growing in the within the gel matrix. Okay. So you can see here something within the gel matrix, and after more time, we see the gel matrix is broken and some crystalline precipitate was observed. So now it is not again gel. So this is a crystalline precipitate. Okay. So what is going on there? So we thought about, we observed that when we take more water, this process is faster. So we studied a time dependent study. In presence of 90%, within 1 hour, so this gel transformed to the crystalline precipitate, this crystalline precipitate. So what we plan, we reduce the solvent water composition. So when we change the water composition, reducing the water composition, we find that 70% gel is stable up to 1 hour, but it is breaking. It is breaking after 2 hours. So we find that 50% gel is stable 24 hours. 50% gel is stable up to 24 hours. When we study in the 40%, up to 1 hour there is no gel, but after 2 hours it is forming gel. So that clearly indicates that when we changing the composition, it actually modulates the lifetime of the gel. So more water, the gel transforms quickly and the gel breaks into the crystalline precipitate. Less water, that is more DMSO is there, so it takes more time to form gel and it is stable up to longer duration. So why we reduce the water percentage? Our planning was to see how the chirality is evolved. So we study the N2 at 45%. Previously, we studied in the 90%. So we saw that in the 30 seconds, after addition of water, we transfer into the CD machine, and we observed that this first peak is slowly growing. Around 10 minutes, it is saturated. It is saturated. But this picture is very nice. But when we see my previous data, it makes me curious because the direction of the N2 molecule in the 45% in the positive direction, but here this N2 molecule in presence of 90%, this is in the negative direction. So I thought about whether I have done everything right. So again we repeated the data and we find that when we study by varying the solvent composition, we find that though there is a chiral molecule, up to 30% of water, it is no CD signal. But after that, we find that up to 55%, the CD signal is in the positive direction. And from 55 to 60, in this range, the CD signal is inverted. And even 90%, we also observe peak in the negative direction. So we thought about what change we have done at here. Basically, we changed the solvent composition to make ensure that this chirality inversion is coming from the solvent. We have prepared the corresponding de-isoleucine. So we have taken isoleucine, we prepared the corresponding derivative, and we term as DN2. And when you study DN2 and N2, we observe that they have mirror image relationship. Okay, so I'm saying it again. So in 50%, the peak in the positive direction for N2, and in the 50%, you can see the peak in the positive direction. When you study in the DN2, 50% peak in the negative direction. And again, when we study in the 60%, you can see the peak, the for N2 peak, the first peak here, this is in the negative direction, like this system. And in case of the DN2, it having the mirror image. But when you only change, all peak pattern are same, only there is a change in the first peak. Okay, so it makes us confirm that the chirality inversion was due to for the solvent composition. And I'm not discussing the other part. So what we observe that this molecule, when we take more water, when you take less water, these gel are stable, and this gel actually forming the right-handed helical nanofiber. And when you took more gel, so it is forming gel quickly, 90% gel. This gel is a transient gel, and it was decomposed to the crystalline precipitate. And when you check the morphology, we observed that is a left-handed helical nanofiber was observed. So in summary, we have discussed the various amino acid based cell assembly system, and in latter part, we have developed NMI conjugated few dipeptide by varying the achiral part, by varying the chirality, and we find uh, how we change the self-assembled chirality. Okay, so in one case we changing the amino acid, changing the achiral chain length, and in third case we have observed by changing the achiral solvent. So I want to thank my student Sobhik Misro. He is my first student. Pijus, just 2 days ago he got his degree. And Somen Kuila, he done the last paper which was published in Small, and this was selected as a frontispiece cover page. Okay, so and they are the two new joining in our lab. So I want to thank University of North Bengal, DST Inspire Fund, SRG grant, and UGC DAE grant. And also I want to thank all the collaborator, and without their help I cannot do work at here. So thank you everybody for listening the talk. I am happy to take few questions.
Thank you, sir, for delivering wonderful lecture on a special topic for all UG and PG students, research scholars, and chemistry enthusiasts. I'm sure that all our participants were thoroughly intrigued by the talk. Now the session is open to questions. I would now request the concerned participants to kindly unmute themselves and ask their questions directly to the speaker. You may also write your queries in the chat box, and I will read out your queries on behalf of you.
quite a few undergraduate students as well with us today. So please don't hesitate to write your
You can ask
questions or ask
question.
your questions. Yeah, I think Dr. Nanda will be happy to answer you all. So please feel free to ask your questions, whatever question you have, undergraduate students as well as postgraduate, whoever. Okay, Bishrit, I think there is one question in the chat box since nobody is.
Yes, sir.
Yeah, please read it out.
Sir, the question is on behalf of NK Ghosh.
Okay.
In slide
Slide
number
number
21, in case of the
21.
self-assembly of naphthalimide peptide hybrid, there are very
Okay.
less hydrophilic groups. So what was the DMSO and water ratio and how the pH was controlled?
Okay. Thank you. So if you see the slide, I'm sharing again. If you see the compound at here, so this in this molecule, this is a hydrophobic molecule. There is the C terminus is protected by the methyl ester group. Okay, so this compound is soluble in DMSO. It is not soluble in water. Okay, so this is a different kind of gelation than the regular gelation. In regular gelation, the compound you dissolve in a one solvent. Okay, either water or organic solvent. In case of the water, you can change the pH. You can add some salt for charge screening to trigger the self assembly. But in this case, this approach is called the anti-solvent gelation. In this case, this compound is not soluble in water. So what the approach is, we dissolve the compound in DMSO and we put water within it. So this is kind of forced gelation. Here there is no effect of pH. So the composition we have varied here, like you can see here, we observe in the 50% DMSO water 50%, DMSO 50 and what is called water 50. In this case, you also observe the gelation for this particular molecule. And in case of the 90% water, even 95% water, you also find the gelation. So 95% is water, 5% is DMSO. So in this condition, we also find this molecule forming gel. If you have any question, NK Ghosh, please ask me directly, so it will be help to explain. NK Ghosh, please, if it's okay, or you can ask me question.
Yeah, if you can please confirm NK Ghosh in the chat box. Are you there? If you can just confirm us. Okay. We had another student, I think Pooja Gain, who had raised her hand.
Yeah,
Yeah,
please.
Pooja Gain, you raised your hand. If you can please unmute yourself and ask the question to sir. You can unmute yourself. You're muted, I think. Okay, maybe some network issue. Okay, in case nobody has any question, Jayanta, I just have just a couple of
Yes, yes,
queries. Yeah.
yes, yes.
So, have you studied the effect of temperature on the stability? Because we know these proteins have got a tendency to denature at certain temperature. Each protein has got its own threshold temperature. So, have you studied the stability of your helical motifs under temperature? Or there is a temperature variable study that has been performed. Any such
Yeah,
denatures?
yeah. Like we have studied, like I have not shown the data at here. Like the last gel, that is in the DMSO water gel. So we have heat the gel material, and we observed that when we heat, the non-covalent interaction are break, and the gel converted into the solution, and when you allow to cool them, so they again self-assemble and they again forming gel.
Okay, so this is a kind of reversible process. So there is no like threshold temperature beyond which, if we heat, there is no chance of getting back and reassembling up to form the.
Like this, these are reversible system. These are reversible
Okay.
system. So if you heat it, so they convert into the monomer, and when you cool down, so when the temperature is reduced, they try to interact with each other, and they are forming the assembly.
Okay. Okay. Thank you. And the other question is regarding this amyloid fibrillation that you're talking about. Now we know there is a class of protein that is the transthyretin protein, which
Okay.
undergoes this kind of amyloidosis, and literature says that they are highly prone to mutations. There have been over, I think, 130 plus mutations that are reported, and there are certain mutations which push the process, which favor the process of amyloid formation, and so they are kind of pathogenic mutations. And then there are certain mutations which are protective clinically, so they prevent the amyloid formation. So in your case, have you like this small peptide sequences that you have taken, are there any naturally reported mutational study on those particular fragments, or how do your introduction of this aryl side chains affect on different mutants of or mutated variants of this particular sequence? Have you done any study of that?
Actually, yeah, I understand the question. Like our plan was whether like I have shown one slide where we have prepared the 4-nitrophenylalanine gel in the DMSO solvent, and if you see in the lower part, I have shown that when we introduce aromatic amino acid, so there is a more intense coloration,
Yes, yes.
more intense coloration that actually indicate that there is a kind of interaction between electron rich aromatic amino acid and electron withdrawing amino acid. So we are basically working on the amino acid and dipeptide system. So we also find in some cases that um, phenylalanine fibril, phenylalanine itself forming fibril. Okay, so this fibril was fibrillation is quenched or is stopped when we introduce certain percentage of 4-nitrophenylalanine in the co-assembly. So we have not gone beyond dipeptide or tripeptide after tripeptide level, large peptide. So we just trying to study in the amino acid dipeptide level, and our plan was if everything is went well, we want to go for the mutation in the large peptide and to study by the co-assembly with the amylogenic or fibril forming peptide whether this mutation whether any effect in the fibril formation. So basically we just started this work, and we observed that aromatic means nitro aromatic amino acid which is electron withdrawing, it has some influence. It can interact with the amino acid dipeptide level, and they have stopped the fibril formation of the aromatic amino acid as well as dipeptide.
Okay, okay, understood. Yeah, because there have been quite a few reports on these mutations, and plenty of studies are going on the effect of single mutant,
Yes,
double
yes, yes.
mutant, triple mutant over this A beta 42 segment.
Mhm.
Anyway, thank you so much, Jantada. If we have any more questions from the
So
participants.
I have seen you have written Professor Bosag is here.
Yes, he was there.
So
He had joined, but I think he might have. Yeah, he joined in between. He was there for some time. He listened to
okay, okay,
your lecture, but maybe, yeah, he has just left now.
okay.
Yeah.
I convey my regards to him and all the faculty member of your college and all the teacher participant.
Thank
So
you.
if you have any other question, I am happy to take.
Yes, UG students, please. We have students from bioscience background, zoology, botany, I think microbiology, biotechnology, biochemistry from different institutes. So please feel free to ask. That will only be the success of this lecture. So please feel free to ask. Faculty members, students, research scholars, please feel free to ask. UG students. I think somebody raised the hand
Hello,
in
sir.
between. Yeah, please.
Sir, I have a question in
Yes,
slide
yes,
number 22 that helicity tuning due change in chiral center.
yes,
Sir, you have shown that variation in alpha carbon center of
yes,
NLV, NDA, and NAA. Sir,
yes,
that
yes,
in NAA,
yes.
sir, chirality is changing, means
N
chirality is breaking down.
NA, I am sharing the slide again. Is visible now?
Yes, sir.
Okay. So what we observe that we have taken the valine, amino acid valine, okay, and in this case, this is the NLV, the L coming from the L amino acid and V coming from the valine. And in the second molecule, this is the NDV, N transform for naphthalimide, D stands for chirality, molecular chirality, and P for valine. Okay. And in the last case, this is achiral molecule. This is two CH3 group is there. So this molecule is achiral. This molecule is achiral. Okay. So we want to check how the molecular chirality have influence on the supramolecular chirality. So we want to check how the molecular chirality have influence on the supramolecular chirality. So for that purpose, we study the gelation, and when we study the gelation, we find that NLB and NDB they are forming gel in that condition. Okay, and due to the naphthalimide group under UV light, you can see this is blue fluorescent. Similarly, you can see here NAA, this is not forming gel, but this is forming kind of assembly. When we check the what is called the CD study, you can see here the NLV study in the 00 means in the DMSO. In the DMSO compound is highly soluble. There is no assembly, but when you put water, it is assembled. So it is exhibiting the CD peak, cotton effect, negative cotton. It is exhibiting negative cotton effect. But when you take NDB, the opposite chirality, you can see it is exhibiting the positive peak. But in the 00%, the line is going through the zero. And NA in the 0% and the 90%, both are passing through the almost zero line. So we have summarized at here that by changing the chirality of the alpha amino acid, we have changed the supramolecular chirality, which is reflected at here. For NLB, it is exhibiting the left-handed helix. For NDB, it is exhibiting the right-handed helix. And NAA, as it is not chiral, so it is exhibiting some kind of flake-like morphology. Okay.
Okay, sir.
Yeah, in case we have no more questions, I think we can end this. Okay, so let me thank everyone. Firstly, I would like to sincerely thank Dr. Jayanta for his excellent deliberation, which I think was highly beneficial to not only the students but all of us, including the faculty members, the research scholars, everyone. So, thank you, sir, for your time and your valuable insights. And I hope with this lecture, many of our students would be encouraged to pursue science, a career in research in future. I would
Yes,
like,
definitely should take. Yes, because in science stream people are coming less.
yes, yes.
So
Yeah.
I don't know how many people will come after NEP in the university. We are getting very less people in the university level actually.
Yes, also at the college level, we are getting very few
Yes,
students.
yes.
But thankfully, like with researchers like you doing such great work, people will be motivated to take up science and pursue a career in scientific research. Thank you again for inspiring and motivating all the students. Uh, I would like to thank our Vice Principal, Sri Shumit Mukhopadhyay, uh, all our faculty members from the Department of Chemistry, and of course our dear participants. Thank you all for being with us. We at Mrinalini Mavavidyapith keep organizing such online lectures, and we will be doing so in near future as well. I hope we will have your active participation in future as well. I'll be sharing the feedback link with all of you in the chat box. Please fill it up, and you'll get an automated certificate that will be generated and shared with you on your registered email ID. So just wait for a second. I'll share the link with all of you. Feedback link. So please make sure that you put your details correctly, because whatever details you'll put in the feedback form, the same will be reflected in your certificate. So it's being shared, the feedback form. Please fill it up and you'll get your certificates. Thank you, Jayantoda, once again. So we will.
Okay, thank you, Prabhuto, um, and all the participant, all the faculty member. I am thankful to the principal of your college and all the faculty member of your department, all the teacher participant who present here, and all the student present here.
Uh, thank you, Jayantoda.
Wait,
I think we just have
sir,
one more
one question
question.
is there
Okay.
Yeah.
from Abu Jafar Sheikh.
Is there
His
any
question
role in?
is there any role aromatic NMI rather than pi stacking?
Yes, mainly this group we introduce for pipe stacking. If you think this NMI group also you can consider as a hydrophobic group also. So this NMI group mainly exhibited the pipe stacking as well as it can also exhibited the hydrophobicity in the molecule.
Thank you,
Abu
sir,
Jahar
for
Sheikh. Yeah, if Abu Jabbar,
clarifying the doubt.
yes, I think his doubt is clarified.
Uh,
Yes, Vishwit, you can officially close the session, but just keep it open for 2 to 3 minutes. I think the feedback form is there, so if participants want, they
The
can fill
form
up.
is not opening actually. Please send the link again.
Let me just check.
It's opening.
It's not opening. Uh, Bishwit, can you just check once? Just click the link and see, since I have already
sir,
done it.
opened.
No, it's not opening, sir.
Me for nobody, it's opening. Okay, okay. Just give me a moment. I'll check. I'll check. Just give me a moment.
I have clicked and it is
It's
opening.
working, John, to the
Yes,
link.
okay.
Okay, okay, okay. Then I don't know why it's not working. Okay, John, to you may leave if you wish. We can
Okay.
sort this out. Thank you
Okay,
again.
thank you, Prabhupada. Thank you.
Thank you, John, to Thank you, thank you.
Thank you, sir.
Okay, let's now deal with this. Not working, sir, but it's working for some. It's not working for some. I don't know why. Let me check. Bishit, is it opening for you?
Yes, sir.
It's opening.
হ্যাঁ এটা ওপেন হচ্ছে তো আমার তো হচ্ছে
তো বাকিদের হচ্ছে না, জাস্ট লেট মি সি।